Wednesday and put it on YouTube. Has anybody checked to see if it's there or not? There should be a new playlist that says spring 2025. So you guys just keep an eye out for that. Okay. This is a mitochondria. Okay. You all have them. You couldn't live without them. They're lovely. They're wonderful. They're great. Before we make all the ATP for ourselves. Okay. Let me ask this question. Anybody have? This is going to sound so. No, I'm just doing a whole thing here all by myself. I'll make sure I ask the question correctly. Anybody? Did your mom like run track or anything? Maybe in high school, or is your mom sort of a pretty reasonable sort of endurance athlete or things? Mostly, I'm having to come to grips with the fact that most of y'all's parents are now about my age, and this is a thing that I have real sort of internal issues about in the midst of that. Right? You guys were all born in like 2002 or 2003 and these kinds of things, and I'm just yes. And my daughter's 6. Anybody have mom who's that way? You got one? Okay. A couple of you guys back there. So remember we asked about who might be the strongest on Wednesday or on Monday, whenever it was. If I want to know who of you guys might have the best potential to be a good endurance athlete, I want to know who had a mom that was a good endurance athlete. Because mitochondria have their own genes. They have separate genes and genetic material from your other cells, and it all comes from your mom. Okay? It all comes from your mom. So if you have a lot of mitochondria in your cells, that likely means that your mom had a lot of mitochondria. And that's one of the genetic determinants of who might be a good or a better, have the capacity to be a good endurance athlete. Okay? So I am one of those lucky people that my mom used to be a marathon runner and do some of those kinds of things. And so, fiber type wise, which is also kind of the bane of my baseball existence, I'm a little bit slower. I do pretty well when I train from an endurance standpoint, but that's the idea behind mitochondria. Okay, we'll talk a whole lot about mitochondria when we get to metabolism. We'll talk about what all the membranes mean. We talk about where all the aerobic processes take place, but I just want you to see these things right now because they are inside of your cells. Okay, I mentioned the other day also about satellite cells. They're muscle stem cells. So they are cells that are going to be adjacent to your muscle fibers that are just sitting there waiting to become new fibers, to repair the existing fibers that we have. Okay, they are going to drive this idea behind protein synthesis, which helps us with hypertrophy. We've not done this in humans, but we do all kinds of stuff in animal models. I can kill off your satellite cells. I can kill off an animal's satellite cells by zapping them with radiation. Kill off their satellite cells, have them do resistance training, muscles don't grow. Okay, muscles don't grow. Some of you have more satellite cells than others. Those of you that have more probably have greater capacity to increase your muscle size. Things like that. Okay. The top part, the things that look like maggots, unfortunately, those are nuclei and satellite cells on the way that those things have been stained out. So you can see that on one fiber, there's going to be a lot of nuclei, there's going to be a lot of different satellite cells to help us repair. Under sort of a light microscope, if you look, you see the nuclei and the satellite cells out on the edge of your fiber. They're really, really difficult to differentiate. The satellite cells are in this kind of double membrane, this little extra pocket where they're going to be there waiting to get activated. Okay. A lot of the hormonal response after a bout of resistance training functions to activate satellite cells. So we'll talk a whole bunch about that in a couple of weeks when we get into resistance training and those things. Okay. So we've covered a lot of stuff about fibers. I want us now to zoom down a little bit closer into inside of a myofibril, which are going to be inside these sarcomeres that are going to be there. So we're going to talk now a little bit about what is a sarcomere
and what's going on with the actin and myosin proteins that make up this sarcomere. Okay, so in our sort of tubes and tubes and tubes system of what a muscle looks like, we have gone from a muscle to a fascicle. Here's a fiber. This is a myofiber. Okay, and a myofibril is just a long chain sarcomeres stacked end to end to end to end. Okay, so we don't really do much at the myofibril level because all of the stuff that happens in a myofibril is really happening within a single sarcomere. Okay, so this picture down here on the bottom left, this is a sarcomere. I'm sure you guys have seen this before. Okay, the kind of reddy pink structures here, those are going to be myosin filaments. And a myosin filament is made up of many, many molecules of the protein myosin. Okay, here is a myosin protein. It has this tail, and then it has these head regions here. Okay, and basically in a thick myosin filament, all of the tails get wound together, and then you have the heads that project off along that at different intervals. And realistically, it's the movement of the myosin head that generates all movement in our body. Okay? These tiny little structures make these tiny little movements, and that's how we get force, and that's how we get movement. Conversely, we have these blue structures, which are thin actin filaments. Actin itself is a sphere. It's a spherical protein. And so we stack many, many of these spheres, these little beads together, and then we wind two strands of them around each other here. Okay, so it looks like this. And then those are going to be also placed in here in our sarcomere in an arrangement that maximizes the ability of us to have an active filament where it can bind to those myosin heads. Okay. On a sarcomere, you guys may have learned in the past, there's different areas and zones and bands and these kinds of things. And that's all great, but all it really is doing is just denoting in the sarcomere where is their actin only, where is their actin and myosin, where's their only myosin. I'm not going to make you guys know all. I do want you to know that the ends of the sarcomere, the boundary at the end of one sarcomere is called the Z line. Okay, the Z line. And there's some other proteins that help anchor all of the other filaments, the actin and myosin filaments, to the Z line. Okay. And so the reason the Z line is important is if you watch a sarcomere when we get a muscle contraction, it's the Z line that appears to move. Sarcomere squishes inward on itself. And so the Z lines go from being out here, and the Z lines start getting squished and pulled in. Okay. And that's going to be the important pieces of all of this. Okay. Don't have questions about that. It's only going to get a little bit more complicated. We're going to dive into a couple of additional structures on here that help impart some of the different functions. Okay. So this is just big picture, right? What that looks like. In a couple of slides, we'll talk about two more proteins that exist on actin. One called troponin, one called tropomyosin. Okay, and then there's this other protein that I said it's a structural protein that I do want you guys to know, and it's called titin. Okay, titin is very interesting, but titin is a protein that anchors, right, our sarcomeres together. It anchors the actin and myosin filaments to the Z line. Titin is arguably like the second or third largest protein in your body. Okay, and it's really cool because about 10 years ago, we figured out that in addition to myosin, titin also has an ability to generate force. Okay, and so for hundreds of years, we thought all force in muscles was generated only by myosin. And we now know that in some circumstances that this titin protein can also help us generate some force. The amount of titin you have in your muscle fibers determined by genetics. Some of you have more titin than others. It's more difficult for us to damage your muscles and make you sore. You may also likely end up being a little bit stronger, all other things being equal because you have more. So this is a big sarcomere. That's what we see there. This is an electron micrograph of individual sarcomeres inside of a myofibril, inside of one muscle fiber. Okay, all of these very nice striped patterns is why we say that skeletal muscle is striating. It has this very distinct striping, which just denotes this nice, tightly organized structure of actin and myosin. Okay, we organize our sarcomeres like this. Everything exists to pack as much actin and myosin into a smaller area as possible so we can get the most force generation that we can get out of sort of that volume or area within a muscle fiber. Okay? So that's why this looks the way that it does. Not all muscle in your body looks like this. Smooth muscle doesn't look like this. The sarcomeres are just a hot mess and are all over the place. And smooth muscle has a different function. It contracts in a different way, but this is what skeletal muscle is going to look like. Okay? To further illustrate the idea that we're packing as much actin and myosin together as we can, this is more of a three-dimensional picture of what those kind of sarcomeres are going to look like. Okay? On the top left, you see what I showed before. Take that sarcomere that's sitting like this and turn it so it's facing you guys. You're looking down the end into it. Okay? And you see an arrangement that looks like this. Okay? Where each of these kind of pink circles is a myosin filament, and then the blue ones are actin. And so what you see is this hexagon shape that is formed around every myosin. There's 6 actin filaments in 3 dimensions that go around. Okay. And if you look on the right, then the reason that becomes important is because all of the actin heads are going to come off at a certain angle. And so along that, the myosin heads, along that myosin filament, you've got heads that are at this angle, right, going along, and you've got some that are pointing straight up and some that are about 2 o'clock, right, and they go around in that direction. And what this does is it allows us to have the most myosin heads bind to the most active filaments as possible in that small space. Okay, in that small space. So again, this is structure, form, anatomy, helping us impart function. Okay, muscles are structured in a certain way from the protein level forward, and that lets us generate the most force we can in the smallest amount of muscle. Okay, that's the idea. Muscle, if you have bigger muscles, you need more energy, which means you need more food. Okay, and at various points, hundreds of thousands of years ago, that could have been a problem for us. We couldn't just run down to Chick-fil-A, right, or run over to Couch and get 14 chicken sandwiches and stash them in our backpack. Right? And so that's why this becomes so important, is that this is sort of form and structure and anatomy imparting function. Okay. Last little bit about all of this, and there's way more on here than you guys are going to have to know, but I couldn't find a better picture that didn't have the extra information. So let me be very clear here, okay? On one myosin head or one myosin molecule, myosin itself is made up of several smaller subunits. We very often call those subunits miromyosin. And so within what we see here, there is heavy miromyosin, there's light miromyosin. That's a way light and heavy denotes how big the subunits are. It's the molecular weight. You just don't have to know all of that. Okay? But the really important thing from an exercise standpoint is there's stuff that happens out here, okay, on the myosin head. The head actually looks like kind of a Y, or it forks like all of this. Okay. And out here on the very ends where the head can attach to actin, there's an enzyme. And that enzyme is called the myosin, that denotes its location, ATPase. ACE means it's an enzyme, and ATPase is an enzyme that breaks ATP down from ATP into ADP and inorganic phosphate. Okay? The myosin ATPase that exists on each myosin molecule comes in several different flavors. And this is what denotes your muscle fiber type, okay, which we'll talk about in a few minutes. People that can run really fast and have fast myosin ATPases, it means that that enzyme cleaves ATP faster, okay, than a slower. And by cleaving it faster, the myosin can move faster. And that's why those people tend to be able to run faster, jump higher. Okay, so this spot, that enzyme right there, okay, is where the majority of energy expenditure from movement in your daily life, it's all coming from the action of that one enzyme on your myosin. So when I have my watch on today, I didn't do this, I meant to, but if you wear a watch or some kind of tracker and you go for a walk, you go for a run, or you go for a bike, and it says you burn 200 calories of energy during that, what that really means is that for the most part, this enzyme on your myosin head turned over enough ATP to be the equivalent of about 200 calories. Okay? When you sprint and you start making lactic acid, things that happen are that we can't make enough ATP to keep that enzyme functioning, and you got to stop. Okay? So it's this one enzyme on a subunit of one protein in a muscle that drives energy expenditure, that drives who can run the fastest and jump the highest, and all of these different things. Who might be a better endurance athlete? It all comes back down to these molecular actions of these tiny, tiny, tiny things inside of the muscle. Okay? That's that big picture. We're going to spend 2 weeks talking about metabolism. And all we're doing in metabolism is figuring out where are we going to get the ATP that this enzyme is, right? And how does that fit into everything else? Additional things, we'll do a lot more about this on Monday, that are on the active molecule, right? Most of you guys are not like up and standing or like having a whole body cramp or anything right now, right? Remember on Wednesday we talked about muscle contractions tend to be voluntary, which means that the normal state of existence in your muscle is not to be contracted. Okay, we accomplish that by having this tropomyosin and troponin protein on your actin coatings. The resting state is not to let myosin bind on to actin. Right? You guys probably maybe you have a younger sibling or cousin around, but the thing at the Black household is we have to have these little child-proof covers and all of the what do you call them, the plugs, right? My daughter loves to plug and unplug things, and if you tell her not to do something, she's just going to try to fucking do it. She's like her mom in that way, which is great for a lot of reasons. She's very confident in herself and has lots of personality, but it can be dangerous. Tropomyosin is a protein that functions like a safety plug on an electrical outlet. It lays over and covers the myosin binding site on actin. And so when myosin tries to bump into it, it can't grab on, it cannot get to the binding site. Okay? So that is there at rest. That's why your muscles aren't contracting all the time. The normal state of existence is can't bind. So troponin is another protein that helps us decide when can we pull the plug out or pull the safety mechanism out of the electrical. And so what you see on here, the darker blue, that's tropomyosin. It lays in the binding site. And then these green molecules are troponin that are gonna be spaced out along there. And when you want to contract your muscle, we send a signal. That signal is calcium. Calcium binds on troponin. The troponin changes its shape, it spins. As it spins, then it just pulls the tropomyosin out of the binding site, and now myosin can bind on. Okay? Then the myosin lets go, the troponin changes its shape back, tropomyosin goes back on there, and now you've relaxed your muscle. Okay? So here again is structure. Proteins that regulate and control when and how we contract our muscle. Can you imagine if it worked the opposite way? If the normal state of existence was, right, this contraction all the time, and we had to send a signal to tell it to stop, you'd spend your entire life being like, don't contract, don't contract, don't contract, okay, now contract, and do all of this. So our ability to have this imparts our ability to be relaxed at rest and then to precisely control how much and when we contract muscles. Okay? How much and when, which is really, really, really important. Okay. So when myosin binds onto actin, which is what you can see in the pictures here, okay? On the left is a resting state. Myosin wants to bind, but it can't because tropomyosin is in the way. On the right, the troponin has moved. We've opened the binding sites. Now the myosin will bind. When actin and myosin bind together, we call that a cross bridge. We call that a cross bridge. We do not get force production until we form a cross bridge. When you guys were looking around thinking who's the strongest person in class, and we were thinking who has the most muscle, what that functionally means is if you have more muscle, you likely have a capacity to make more cross bridges and therefore generate force. Okay? So we get that binding, we form a cross bridge, and then what happens next is why we get moved. It binds, and then the myosin pulls the actin filament along, or it attempts to. It pulls it. That's where we get force. That's where we get movement. That act of trying to pull is called a power stroke. Okay? It's called the power stroke. Binds, power stroke, and it moves. Then it lets go. It goes back and it tries to bind again, and it pulls again, and it lets go. And it can do that hundreds of times. I mean, not hundreds, but tens of 20s of times in a second. Okay? So when you do a full muscle contraction, what you're getting is thousands and thousands of individual power strokes over and over again down on the molecular level, at the level of the cross bridge. Okay? Having that power stroke, pulling that actin molecule, okay, getting movement of the sarcomere, which then makes the myofibril move and the fiber move. This process is often referred to as the sliding filament theory of muscle contractions. Okay, you make a cross bridge, it pulls on the actin slides, okay, and pulls the sarcomere together. It pulls those Z lines together as the filaments slide by each other, and voila, we get moved. Okay, that's what's going on. So you do that, and it takes, milliseconds for all of that to happen once, and you don't move very far. Imperceptible. You get one cross bridge cycle or one sliding filament from one myosin thing, and you'll never know that you're having any movement at all. Okay? So in order to get a contraction, in order to get movement, we must do bind, cross bridge, power stroke, let go, bind, power stroke, let go, over and over and over and over again. Okay? And so that process where we go from binding to tilting the head and getting a power stroke to letting go to then putting the myosin back where it needs to be so it can bind again is called the cross bridge cycle. Okay, that's called the cross bridge cycle. I need you guys to memorize this as it is written on this slide. Okay, I'm sorry, but I just need you to do that. Okay, we're going to start at the top where you have a resting muscle, no contraction whatsoever. Okay, tropomyosin is sitting in the binding site. We're ready. Okay, you all think contract, right? Calcium comes in, binds on the troponin. We move to tropomyosin. So then we get the binding site open, and then we get to this step here. Myosin binds. We have a cross bridge. Okay, we have a cross bridge. It's important at this point for me to point out what is happening with ATP and ADP and inorganic phosphate as it relates to the myosin head. It's really important. Okay. At rest, before you form a cross bridge, every myosin head has a molecule of ADP attached to it and a molecule of inorganic phosphate attached to it. Okay, that's what we see at the top. When we make a cross bridge, the first thing that happens is we release that molecule of inorganic phosphate. Okay, that triggers then the power stroke. That triggers move and tilt the myosin head. It tilts, then we release the ADP. Right? Then we release the ADP. We call this top part where both of them are bound, we call that a high energy state. When we get down here and they've both been released, we call that a low energy state. Okay. So we have now gotten a power stroke. We've slid the actin, so we've gotten a little bit of movement. And here's one of the most backwards things to me in all of physiology, conceptually. Okay. You have generated force, you have generated movement, and yet you've actually used no energy yet in this entire process. Okay? You do not use energy to get a power stroke to happen. So that again, do not use energy to make a power stroke happen. So we don't use energy to get force or the initial movement. You guys are thinking, well, that doesn't make any sense, right? So what happens next is we're stuck, okay? The myosin is bound and it waits. And ATP comes in and an ATP molecule binds to the myosin, and once ATP binds, that triggers the myosin to let go of the actin. That's the signal. Okay? We've now broken the cross bridge, we've let go. We've relaxed that cross bridge. Then the myosin ATPase enzyme takes that ATP molecule and it splits the phosphate off. Okay? That process, that hydrolyzing that ATP, now we've used energy. Okay? And what happens is it's broken into ADP and inorganic phosphate, both of which stay attached, but that process of splitting it takes the myosin head and basically cocks it back to where it needs to be so that it can bind again. Okay? When we do a muscle contraction, we use a shit ton of energy. But the energy is used to let the actin and myosin let go of each other, so that we can then initiate the next cross bridge cycle. Okay, so we're actually using energy to get a muscle to turn off. And as you go through a full range of motion, every cross bridge that is in your muscle, which there might be millions, every one of them goes through this process of bind, generate force, let go. Bind again, generate force, let go. Okay, over and over and over. And that's the cross bridge cycle.
Okay? So it's always seemed backwards to me that we actually use energy to get it to turn off rather than to generate force. I promise you, that will be a test question. Okay? That will be a test question. Okay.
How are we doing on time? Are we getting close? We're right at. Okay. Covered 5 whole slides.
All right, questions? No? Okay, please make sure that you're working on the Connect assignments if you don't have those. Okay, I will see you guys on Monday. It's Friday, here's your weekly. I won't see you on Monday. I will see you next Wednesday. You get a long weekend. Make good choices. All thanks.
That's a good place to stop. You're about to go into front.